All right, guys, this is the first video lecture on Chapter 14, Mendel and the Gene Idea. This is all about genetics. The reason why you find genetics, or the genetics chapter like this, always listed in textbooks right after the chapter on meiosis, Chapter 13 in our case, is because many of the patterns of heredity that we see in genetics are a result of the way that chromosomes behave during meiosis. So there's a link there. All right, so we're going to spend the first, half or so of this chapter kind of talking about Gregor Mendel and the research that he did back in the 1860s, because the results that he got from breeding pea plants in his garden sort of form the basis for modern genetics. The work that he did was very important. And no one knew a lot about heredity or genetics in Mendel's time, but he was sort of a visionary person. He was doing what for the time was actually very cutting-edge research into some very important questions about genetics and heredity. For example, one question that was very important at the time is what principles actually account for the passing of traits from parents to offspring? How is this controlled? How is it regulated? Why do we see, in some cases, children being a nice mix of both parents' features, but in other cases we see children that look to get all their characteristics and traits from just one parent only? And those are just some examples of genetic patterns that we see. So a major question then was, what principles account for the passing of traits from parents to offspring? This was a major question in Gregor Mendel's time. Well, prior to Gregor Mendel's work, the overriding idea, the most widely held belief at the time, was called the blending hypothesis for how heredity worked. And the blending hypothesis is the idea that genetic material from two parents literally blends together in the offspring. Sort of like, for example, if you mix blue and yellow paint, you'd get green, right? Is it then possible to separate the blue and yellow back out of the green? No, they're then inextricably mixed together into a different color, green. That's how people viewed heredity. They viewed, whatever information was coming from the father and the mother as uniting and mixing together inextricably in the offspring. And that's why, in very many cases, offspring appear to be a nice mixture of both parents' traits. Well, we know that that idea is wrong. And the reason it's wrong, and this was actually a flaw that was noted at the time, but there was no better explanation until Gregor Mendel did his work. The reason we know that the blending hypothesis is wrong is because over time, blending would tend to reduce genetic variability and physical variability between individuals. So think about it this way. Let's say you have a bunch of buckets of red and white paint. Maybe the red paint buckets, represent the fathers, and the white paint buckets represent the mothers. If you get a white bucket and a red bucket that mate, imagine pouring half of each of them into a new gallon bucket. You would get half red, half white, mix them together, you get pink, right? So all of the offspring in the first generation would be pink. That would be their genetic makeup, quote. Okay, now, those individuals start to mate with each other. So you get pink with pink. Then in this second generation, what do you get? Just more pink. And so on down the line. Blending tends to make everyone the same. It tends to homogenize the differences. And so this didn't jive with what people visually observed, which is every generation you get new individuals that look very different from each other, very different from their parents, very different from the prior generation, and so on down the line. So blending hypothesis didn't make a lot of sense, but they stuck with it until Gregor Mendel came along because nobody had a better explanation. So with Gregor Mendel's work, as a result of his work and his results, he developed what's called the particulate hypothesis. So let me write that down. Particulate hypothesis. This is the idea that parents pass on discrete heritable units, or particles, so to speak, what we would today call genes, okay? And basically the idea is that you get certain heritable factors or units, genes, from one parent and also from the other parent. And yes, they do come together in your cells, but they don't mix together. They remain separate, distinguishable biological entities, okay? Your father's gene on one chromosome, your mother's gene for that same trait on a separate, distinct chromosome. That's the idea behind the particulate hypothesis. And Mendel developed this idea through very, very basic and very simple experiments with garden pea plants. Okay, so Gregor Mendel was a monk, all right? This is Gregor Mendel right there. You see, you can almost see, he's holding up a plant of some sort. He was actually, fuchsia, I think it is. He was actually trained as a botanist. He had a 4-year degree in botany. So he was a biologist. And he was living in this abbey with his other monks in what would become Czechoslovakia. Today Slovakia, I believe. And they had a large abbey garden where they grew a lot of their food. Well, Gregor Mendel decided to do some experiments with pea plants, some genetic experiments. And he used the abbey garden to do this. Okay. All right. Based on these experiments, which I'm getting ready to describe, he discovered the basic principles of heredity through these very carefully planned, yet really very basic experiments, as we'll see. Now, Mendel was a really smart guy. There are some people that we look at today, like Stephen Hawking, Charles Darwin, Albert Einstein, for example, Richard Feynman, and we think of as genius, right? Because they could see things that other people just couldn't see, very, very intelligent, and they contributed so much to society through their scientific research. Well, when you think of genius, Gregor Mendel is not one of those people that pops into mind, but I actually think he was a genius because you have to realize the environment he was working in and the lack of understanding that they had of biology back in the 1860s. DNA wasn't even discovered as a component of our cells until the 1890s. No one knew what chromosomes were. They'd just barely been visualized with rudimentary microscopes they had of the day, but no one knew what they were doing in cells. The lack of knowledge was just astounding compared to what we know about today. Yet, in spite of that lack of knowledge, Gregor Mendel was able to see things and make logical deductions. He was able to cross conceptual gaps that I think are just astounding for the amount of information he had available to him. I think he was a genius, and I don't think he gets enough credit for that. Okay, so Gregor Mendel's approach allowed him to deduce the principles that had remained elusive to others. Okay, this is him seeing things that just other people missed. Let's define a few things here first. So, first of all, character. Okay, what is a character? A character is a heritable feature that varies between individuals. So, flower color, for example, is a character. Stature can be a character. Skin tone can be a character, all right? Now, those characters can come in different forms or different variants. The variants of characters are called traits. So let's say we're talking about eye color as a character. Blue eye color is a trait of that character. Brown eye color, hazel eye color, green eye color. Those are traits of the character that is eye color, okay? Trait is more specific. It's a variant of the more general character, all right? Now, Gregor Mendel, again, he was a botanist, so he was interested in plants. He decided to use pea plants for his genetic experiments, and they were available to him in a wide variety of different forms or different varieties. There were basically 7 different varieties that he used, and we'll see what those varieties are in a little bit. So that was one advantage, the fact that they came in different varieties. He could look at 7 different characters, in other words, that came in different traits. That was one advantage to using these pea plants. What was another advantage? Well, they had a short generation time. He could plant, a row of pea plants, and in a matter of a month or so, those pea plants could be reproducing and making the next generation. So he could get 3 or 4 generations in one growing season, if he really tried. Each plant also, that was one advantage, each plant also, as a second advantage, could produce a large number of offspring. So think about a pea pod, right? A pea pod may contain 5, 6, 8 peas, whatever. The pea is the seed. It contains an embryo for a pea plant. You plant that pea as a seed, it has the potential to grow up into a new plant. Well, on a single plant, you may have 50 pods. So if you have, let's say, just 6 peas per pod on average, That's 300 potential offspring from that one plant. So the large number of offspring meant that he could see a wide swath of the potential variability in the genetic system for whatever character he was looking at. All right. Third, or the next major advantage here of using pea plants, is that he could very precisely control reproduction between plants. Okay, he could control what plants mated with what plants, and he could therefore know who the parents were of particular offspring. That's important for comparison, to be able to see how certain traits of a character are passed down from parent to offspring. The reason he was able to do this is because pea plants are flowering plants. Okay, flowering plants, in many cases, can either self-pollinate or they can be cross-pollinated. So let's take a look at this figure here to show you what I mean. All right, this is a flower of a pea plant. Okay, what you have in that flower, and again, the flower is the reproductive structure, is both male and female structures. So you look at this vase-like structure I'm showing you here. That is the female portion of the plant. The female reproductive structure is called the carpel. The eggs are housed down in the vase-like structure of that. All right. Now, let's go and look at these over here. These little spindly structures that you see, those are the stamens. They are the male portion of the plant. They are what produces the sperm. Okay, plant pollen is sperm. If you've seen these on an orchid or something, rubbed your finger across them, you come off with that sort of powdery stuff on your fingertip. That's the pollen. Okay, those stamens produce that. Well, so a flower has both female and male structures. That means that the stamens in this flower could produce pollen that would pollinate the eggs down inside the carpel of that same flower. That's self-pollination. Well, Mendel didn't want that to happen. He wanted to have crossing between two different individual parents, so he would actually take a pair of scissors and remove the male stamens, leaving only the female carpel in that flower. So if these plants, where he had done this, were going to be pollinated, it would have to be with pollen from a different plant. So he would collect pollen on a brush from a white-flowered plant and dust it onto the carpel of a purple-flowered plant. He would therefore know which two plants were the parents. They were called the pea generation. That carpel then, once fertilization had occurred deep inside of it, would grow into a pea pod with the individual peas inside, as you can see. He would then plant those peas and produce the F1 generation. These are the offspring of these two parents. All right, and this is actually showing you one of his first results. Even though both parents were different colored flowers, purple versus white, all of the offspring in the F1 had purple flowers. That was an interesting result to him. The question was, why are they all purple? Why are there no white flowers in these F1 offspring? We'll see the answer for that a little bit later on. Okay, now, an important point, or an important thing, a decision that I should say that Gregor Mendel made early on, is that he chose to track only characters in his pea plants that were what he called true breeding. All right, true breeding. So what is true breeding? Okay, well, go back to the flowering plants. I said they can self-pollinate, right? So let's say you have a pea plant, a single pea plant, and the flowers in it undergo self-pollination, meaning that the pollen from a flower fertilizes the egg inside that same flower. All right. If for that character, let's say flower color, this plant is true breeding, then all of those offspring would look just like the parent. All of the offspring as a result of self-pollination would look just like the parent for that character. We would then say that because the parent was purple flowered, let's say, and the offspring resulting from self-pollination were also always purple flowered, and when they self-pollinated, they also always make purple flowered offspring, we would say that purple color, or purple as a trait of flower color, breeds true. All right, that was an important decision that he made very early on. Okay, we'll see from a genetic standpoint a little bit later on, maybe in the next video, what true breeding actually means. All right, so what was a typical experiment that Mendel did? Well, he was carrying out what were called, or what he called, hybridization experiments, okay? In a typical experiment, hybridization experiment, Mendel would mate two contrasting true breeding varieties, okay? So for example, purple flowered plant and a white flowered plant. So he's looking at the character of flower color, but he's hybridizing two parents that contrast in that trait, purple versus white, for example. Okay, so that's the parent generation. He would then conduct his experiment like I explained before, and he would produce the F1 generation. He would then look at the distribution of that trait, flower color, let's say, in the F1s and compare that to the parents. He would also then either allow the F1 offspring to self-pollinate or cross-pollinate with each other to produce the F2 generation, which would be the grandchildren, so to speak, of the original parents, to see what would happen with that trait in the F2 generation. All right. So by doing this, Gregor Mendel developed what he called his law of segregation. Okay. Gregor Mendel actually developed two laws. The first one is the law of segregation. All right. And I'll explain what this means here in just a minute. But he developed this law of segregation by doing his very first, most simple experiments. Okay. So when Mendel crossed contrasting, true breeding, white and purple flowered pea plants, all of the F1 hybrids were purple, as you saw in that previous figure. Okay. Well, again, he asked the question, why are they all purple? Why no white? When Mendel crossed the F1 hybrids that were all purple, many of the F2s that came from them had purple flowers, but some had white. So white showed up again, all right? So white appeared to skip a generation then. Mendel discovered a ratio of about 3-to-1 purple to white flowers in the F2 generation. All right, that's what you see here. So here we have Mendel's experiment. This is one of his most basic, simple experiments, first experiments. You see he has a true breeding purple flowered plant, true breeding white flowered. Again, if this one self-pollinates, always makes white. This one self-pollinates, always makes purple. That's true breeding. When he crosses them through a hybridization experiment, he makes F1 hybrids, and he found they were all purple in every case. All right. He allows these to self-pollinate or cross-pollinate with each other, and he got the F2, and he found typically 3-to-1 purple to white. So two things here. First of all, white shows up again. Secondly, it was always in a ratio of 3 purple to 1 white. So 705 purple flowered plants to 224 white flowered. That was an actual result. Look at these numbers. These are numbers of plants. This is one man in the 1860s with no one helping him, no computers. He has an abacus, probably a quill pen and parchment paper, and he's doing all this work. Yeah. All right. That was his first experiment. Okay, so he got this 3-to-1 ratio. This was just in one character, flower color. There were 6 other characters he looked at. And every time he did this experiment in those other 6 characters, he always got this result. The F1s always looked just like one parent. In the F2s, mostly one parent, but some looked like the other parent, so he had the second character show up when it wasn't present in the first generation. This result happened across the board. So he knew that it wasn't something that was just sort of a fluke of one of these particular characters he was looking at. It was the way that the genetics actually worked in these organisms. Okay, so with this result, Mendel reasoned that only the purple flower factor, as he called it, today we would call it a gene, that only the purple flowered factor was affecting flower color in the F1 hybrids. All right. The factor for white, Mendel called the purple flower color a dominant trait and the white flower color a recessive because in the F1s, white didn't show up. Purple did. So purple was dominant, white was recessive. Okay. Well, he knew that the white color factor gene, let's say, was present in these individuals because if it weren't, it could not possibly have shown up again in the F2s when they reproduced. So the white is there, but it's not being expressed. It's not showing up. It's being dominated by purple for some reason. Okay, so that's an important result. This is where he came up with the idea of dominance and recessiveness. Right? Okay, And again, as I said, he observed the same pattern of inheritance in 6 other pea plant characters, each represented by 2 traits. So again, what Mendel called a heritable factor is what today we would call a gene. All right, remember that. Okay, let's look at the other traits or characters he looked at rather. So we've talked about flower color. Oops, sorry. We've talked about flower color, purple versus white. We saw the ratio of 3 to 1 in the F2s, okay? Look at seed color, yellow versus green. Look at the ratio, 3 to 1, roughly. Seed shape, round, wrinkled, 3 to 1. So you see a pod shape, pod color, flower position, stem length. In every case, he sees roughly a 3 to 1 ratio. So again, this is how he knew that what he saw in his flower color character experiment, purple versus white, was not just a fluke unique to that particular character. It was something systemic in the way that the genetics works, because he saw it across all 7 different characters. All right. So he gets these results. He develops basically a model to explain why he's seeing these results. Think of a model as a hypothesis, all right? So he developed this hypothesis to explain the 3 to 1 inheritance pattern that he observed in the F2 offspring, but also to explain why it is in the F1s only 1 trait showed up. All right. There are 4 related concepts that make up this model or this hypothesis to explain what's going on here, okay? And today, these concepts can be related to what we know about genes and chromosomes and in particular, what goes on during meiosis. Gregor Mendel knew nothing about meiosis. No one really understood meiosis at the time. So when he developed this hypothesis, this model to explain these inheritance patterns he's seeing in his pea plants, he is indirectly describing what's going on in meiosis without ever having observed the process. Okay. All right. So the first concept that he rolled into this overall explanation or model for what was going on here is that there are alternative versions of genes, okay? What we call alleles. Alleles are alternative versions of genes. Let's say we have a gene for flower color in pea plants. One version or allele codes for purple. The other version or allele codes for white. That's all it is, okay? An allele is just a different version of the same gene. All right, so we have 2 different alleles then for flower color, all right? And they're alternative versions. Now, each gene also, for a given character, resides at a specific location on a chromosome. The location where a gene is found is called its locus. Think of the locus as literally its mailing address, its physical location, all right? And every gene in every organism always has the same locus. So the gene for flower color is found in one spot on chromosome 3, let's say, in peas. Okay, it's always found there. The gene for pod shape, let's say, is always found maybe on chromosome 5 in a certain spot. That's its locus. Okay, all right. So let's take a look at that. So here we have, let's say, the gene for flower color. All right. So we have 2 chromosomes here. Remember, we're talking about diploid organisms here that have 2 copies of each chromosome, therefore 2 copies of each gene. And so we have our gene for flower color at that point on that chromosome. That's its locus. So this individual has 2 copies of that chromosome, therefore 2 copies of that gene at that locus. It has a purple variant or allele and a white allele. All right, what is the difference between these 2 alleles? Why do we have different versions of genes? It's due to past mutation. A mutation is just an error that occurs when DNA is being replicated prior to cell division. So back in S phase of the cell cycle, like we talked about. All right. So let's say we have the purple allele, which has this sequence, C, T, A, A, and so on, right? So let's say that at some point in the past, that C mutates. We get a typo when this sequence is being copied, and now we get an A there instead. That's our mutation. That makes a new version of that gene, a new allele, and that one letter change in that base could result in a gene that codes for white now. All right, well, let's see why these genes code for different colors. Well, this gene codes for a protein. It's an enzyme. And that enzyme helps to build purple pigment, which then gets deposited in the tissues of the flower, and we see that as purple. The mutated version of the gene, the white allele, does not actually result in an enzyme. No enzyme is produced from that gene. So there's not an enzyme present to build purple pigment, so you don't get pigment put into the flower as a result. Now, this individual has a purple flower color allele and a white, so it does actually get purple pigment, and you see it as purple. But what if we had two white alleles, one there and one there? Well, then neither gene would produce white pigment, or the enzyme. You wouldn't get purple pigment, and so you would see the flower as white. All right. Second point, or second part of Gregor Mendel's explanation. The first was that we have alternative versions of genes called alleles. The second is that for each character, an organism inherits two alleles, right? We're talking about diploid organisms. So two alleles per character. Okay. One from each parent. Mendel made this deduction without knowing about chromosomes, without knowing about meiosis, any of the stuff we talked about in chapter 12 and chapter 13. Okay, again, smart guy. The two alleles at a particular locus may be identical, as in the true breeding plants of Mendel's P generation, or they could be different. In the previous figure, I showed you a flower or a plant that had a white allele and a purple. They can be different like that. I also said they could both be white, but they could also both be purple. It just depends, all right, on what they get, on what that individual gets from its parents. All right. So, what is true breeding? I said that a true breeding individual, when it self-pollinates, produces offspring just like itself. Why does it do that? Because it has two copies of the same allele. So a pea plant that is true breeding for purple flower color has two copies of the purple allele. One that breeds true for white has two copies of the white allele. All right, keep that in mind. We'll see more about that a little bit later on. Okay, third point, or third part of Gregor Mendel's explanation for these patterns of heredity he's seeing in his pea plants is that if the two alleles at a locus differ, then one of them, the dominant, determines the organism's appearance, and the other, the recessive, has no noticeable effect on the appearance. All right, that's his third point. If the two alleles are different, one of them will determine the appearance, flower color, let's say, the other one will not. So in the flower color example, F1 plants had purple flowers because the allele for that trait is dominant. The allele for purple is dominant, and they all got one copy of that from one parent. All right. Fourth point, or part of his explanation for these patterns of heredity, is actually the law of segregation, as I said before. Okay. It says that the two alleles for a heritable character separate or segregate during meiosis, during gamete formation, and end up in different gametes. The law of segregation is talking about the way that the chromatids, sister chromatids, separate from each other and end up in different gametes in meiosis. It's talking about going from a diploid cell to, let's say, 4 haploid sperm cells. Okay, that's law of segregation. So an egg or a sperm gets only one of the two alleles that are present in the parent organism. So this segregation of alleles then corresponds to the distribution of homologous chromosomes to different gametes in meiosis. All right. Remember, we started off in meiosis, let's say, with a homologous pair of chromosomes. You have 4 chromatids there. And in your first cell division, we have 2 chromatids on one chromosome going to one cell, 2 on the other going to the other cell. That's meiosis 1, right? And then in meiosis 2, we have, in the case of sperm production anyway, we have 4 cells, each one getting only one of those original 4 chromatids. Okay, so these guys are diploid, these guys are haploid. All right, Gregor Mendel had not seen that process of meiosis under a microscope. It wasn't understood during his day, but he figured out this is what's going on based on just looking at patterns of characters, physical characters from one generation to the next. All right. This model then that Gregor Mendel developed accounts for that 3-to-1 ratio that he saw in his F2 generation of crosses, okay? Mendel developed a very handy tool called a Punnett square to show what the results of a hybridization experiment or a hybridization cross would be. And in this case, an uppercase letter represents a dominant allele and a lowercase represents a recessive. All right, let's take a look. So this is Gregor Mendel's first experiment with pea plants or flower color in pea plants using the Punnett square now. All right, so remember he had two true breeding parents. Okay, one that had 2 copies of the dominant allele, uppercase P, one that had 2 copies of the recessive, lowercase p. Remember, in this case, white is recessive, purple is dominant. So when these individuals make gametes, sperm and egg, each gamete only gets one of the 2 alleles because gametes are haploid, right? This parent only has dominants, so its eggs, let's say, are all dominant. This one only has recessives, so its sperm, let's say, are all recessive. So a dominant and recessive come together in the F1s. The F1s then have 1 dominant and 1 recessive. So because dominant is purple, we see them all as purple. That's why all the F1s are purple. They had 1 copy of that dominant, and that's all it takes for flower color to make them purple. All right. Now, when these guys undergo meiosis to make their gametes and either self-pollinate or cross-pollinate, now half their gametes have the dominant and half have the recessive because they have 1 dominant and 1 recessive. All right. Now, this is our Punnett square down here. It shows us all possible combinations of our parent gametes. So let's say we have the father's gametes here. Half of his sperm are dominant, half are recessive. The mother's gametes, half the eggs are dominant, half recessive. If this egg and that sperm unite in fertilization, we get an individual that's purple because it has 2 dominant alleles. However, notice here, if this sperm fertilizes that egg, we have an individual that has 1 dominant and 1 recessive. But again, that gives you purple because it has a dominant. The recessive has no effect. Same thing here, just different way to get this. We have a dominant sperm fertilizing a recessive egg. Again, dominant recessive, it looks purple. The only way you get a white offspring is if you get 2 recessives. So a recessive sperm fertilizing recessive egg, 2 recessives, that gives you a white. 3 purples, 1 white. That's his 3-to-1 ratio. That's exactly why he saw that 3-to-1 ratio. He worked it out, and logic told him this was the explanation, and it's been confirmed ever since. This is exactly right. He nailed it. Okay? All right. So, some terms. We just saw 3 different possible allele combinations. Okay, I'm going to use the letter A, for example. We saw an individual that had 2 copies of the dominant. We saw one that had a dominant and a recessive, and one that had 2 recessives. All right. Remember, this one was purple, right? As was this one, and this one was white. Okay. What do we call these? Well, first of all, this one, 2 dominants, and this one, 2 recessives, they are true breeding, okay? This one with dominant and recessive is not true breeding, okay? Genetically, what do we call these in terms of modern genetics? Well, we call this one homozygous dominant. So homozygous means the 2 alleles are the same, and dominant meaning they're both dominant. We call this one homozygous recessive. Okay, again, homozygous, they're the same, both recessive. This one, however, where we have a dominant and a recessive, it's not homozygous. We call it heterozygous. And it's not heterozygous dominant, heterozygous recessive, it's just heterozygous. So these 3 terms then are what I'll be referring to from now on, and you should know that this is what they mean. Okay? All right. So homozygotes then are true breeding, heterozygotes are not. All right, two other very important terms. First of all, we have what's called genotype. The genotype is simply the genetic makeup, all right? So homozygous dominant, homozygous recessive, heterozygous, those are genotypes. They are the genetic hardwiring, okay? The genotype largely determines what we call the phenotype, the physical result, or the physical appearance, if you will, of the genotype. So we have a homozygous dominant pea plant rather, that's purple. So this is our genotype, homozygous dominant. The phenotype is purple. Genotype here is heterozygous, phenotype purple. Genotype here, homozygous recessive, phenotype white. All right? Okay. So let's take a look at Mendel's results. So that 3-to-1 ratio I talked about that he found in his first experiment, that was called a phenotypic ratio. It's the ratio of one phenotype, purple, to the other, white. So 3 to 1. That was a 3-to-1 phenotypic ratio. But we can also look at that in terms of a genotypic ratio. The genotypic ratio is the ratio of genotypes to each other. So if you remember back to the Punnett square, go back for a second, okay? We had one homozygous dominant, two heterozygous, to one homozygous recessive, all right? That's our genotypic ratio. So one homozygous dominant, two heterozygous, to one homozygous recessive. The reason why we have this difference is because you can imagine if you're just looking at phenotypes and you see these three purple flowered plants, you don't know just from physical appearance what their genotype is. They could be homozygous or heterozygous because both of those genotypes give you the same phenotype, purple. What you do know is that if you see a white flower plant, it's homozygous recessive. It has to be. That's the only way you get a homozygous, or that's the only way you get a white flowered plant, all right? Okay, so a phenotypic ratio then is 3 to 1, whereas our genotypic ratio is 1 to 2 to 1, okay? All right, I think that is a good place to stop for this first video. I'll pick up with Mendel's second law, which is the law of independent assortment in the next